telomeres are going to involve in the end region of a chromosome. It is very important for the stability of a chromosome, stability of the nucleic acid. Telomeres are important. Therefore, to protect the chromosomal deterioration, after certain cycles of replication, a nucleic acid gets deteriorated at the end. The end of a nucleic acid to be deteriorated or protected by these telomeres. So, in the chromosome at the end region is known as telomeres, which consists of repeated units of nucleic acid. These repeated units of nucleic acid are going to protect whole DNA, which is going to protect it from deterioration during the cycle of replication. Every cycle of replication causes a loss of certain nucleic acid. Therefore, this loss is going to be taken up by these telomeres, by the sequence known as telomeres. Therefore, does not affect the nucleic acid to be coded for the RNA or protein. Next, we are going to see about the non-coded functional RNA. From this of all, we also see the non-coding DNA, which is also going to be functional, but its function is going to not a major function. It's a major function. It's going to be starting the DNA. It can be an enhancer or inhibiting region, or it can be a promoter, like that. But it is not directly involved. It is involved as indirectly. But it can be present in the DNA, but it not converted into a RNA. A certain genes which is going to be present in the DNA but not converted into a RNA for the protein synthesis is known as introns. But there is a functional RNA, non-coding functional RNA and non-coding non-functional RNA. These like promoters, enhancers are known as present in the DNA, not converted into a RNA. But some of the fragments of genes are converted into a RNA but not involved in protein synthesis. These are known as non-coding functional RNA. They are converted into RNA but not converted into a protein. Where before we saw non-coding DNA, introns, these introns are present in the gene of DNA, not converted even into a RNA. This is the differences between these introns and non-coding functional RNA. So, these are functional RNA molecules but not translated into proteins. But before that we saw introns. They are functional but not even converted into a RNA. So, these non-coding functional RNA examples are rRNA, mRNA, tRNA. Where mRNA is going to be single stranded, but rRNA can be a double stranded too, due to its tertiary structure, looping and hydrogen bonding, which causes its rRNA to be double bonded too. But mRNA can't be a double bonded at any of the cases. But tRNA is known to be present as a clover leaf-like structure. These tRNA and rRNA to be involved in translation process. But mRNA can be present in the exon formation. mRNA is a exon which is going to involve in transcription. There is also a microRNA. A microRNA is also present, which is also consists of certain function. It is also a single stranded one, which consists of small fragments of DNA which is converted into a RNA. Next, we are going to see about the cis-trans regulatory elements. This cis-trans regulatory elements is very important because based on its name, it differs. Where the cis regulatory elements or the sequence which is going to near the transcription region of starting site, whereas trans regulatory element is also going to control the gene expression, but it is found to be at the distance region. Therefore, these both are going to control or regulate the transcription, but based on its location, these changes. Thus, this is going to be present near the transcription region, where trans is going to be present at the distance from the transcription region. Both are going to regulate the transcription. So these are the introns to be present.